Welcome to the second part of this case study where we look at a cancer drug target discovery pipeline that is based on an RNA-Seq workflow. As part of this workflow, we're comparing tumor and healthy tissue gene expression profiles in order to identify genes which are differentially expressed in a tumor that could make up interesting potential drug targets for oncology. The first part of this case study, we looked at how to translate the research process into a data structure that defines the relationship between the projects, the samples, and the raw data that are produced here, how to translate the data structure into a relational database scheme, and how we can access our research data via a Streamlit user interface that allows us to manage these data. What we'll look at now is basically how we can go from the actual raw data or FASTQ files to the differential gene expression, which involves the bioinformatics steps that we need to analyze our data. And we will also see how we can integrate the data analysis steps also in our user interface and explore the results. So the data analysis is made up of 4 steps that start with uploading the FASTQ files into an input S3 bucket until getting a list of differentially expressed genes. And the first 2 steps are the FASTQ registration and the FASTQ QC steps. All of the steps are implemented as Docker containers, which makes this pipeline kind of flexible, so individual steps can be upgraded or extended without the need to reorganize the whole pipeline. And each of the containers is storing its results in the backend so that everything is structured and stored and can be accessed. The first 2 steps are related to the FASTQ files. First, the FASTQ files are registered. This means that the format is checked, and if it's a valid FASTQ file and the read1 and read2 files are paired as data set, then the FASTQ files are stored in our database. Then the second step involves the FastQC, a tool. I think many of you know this. It's a standard tool to check the read quality of a sequencing run. And this is executed for each FASTQ file, and then the results are also stored. Both of these steps are actually chained by an AWS step function. This is a very elegant way of sequentially running Docker containers on AWS based on an input trigger. In this case, our input is a new FASTQ file which is uploaded, which will then automatically trigger these functions. So all of this is happening in the background of our data platform. Then after we performed this FASTQ registration, we have an RNA-Seq processing step, which is kind of the core of each RNA-Seq bioinformatics. We need to perform the read alignment, so we need to match the individual reads to genome and quantify genes and transcripts that we can then use for actually performing the differential gene expression. And in this case, we use Callisto for RNA-Seq processing, which is a super fast pseudo-alignment tool that can analyze large numbers of samples in relatively short time with limited resources. And we built a container here that runs Callisto, and we registered this container using AWS Batch. AWS Batch, again, is a great tool to perform parallel batch jobs. On AWS, we can provide the maximum number of allowed resources, and then AWS will basically scale the data analysis in parallel for us. So we can run tens, hundreds of samples in parallel and like this build very fast and super scalable pipelines. After we performed the RNA-Seq processing, we need to perform the differential gene expression. For this, we use the rsleuth package that comes from that comes together with Callisto and is meant for using Callisto outputs and perform differential gene expression on the gene quantification. And we embed sleuth in an R runtime container. And in this case, we run this container as a Lambda function. This makes the whole process much faster, and we can actually trigger the differential gene expression from our backend and just need to wait for a couple of seconds, and then we get results from the comparison we want to make. Those are the 4 steps that make up the bioinformatic processing. Let's have a look how to deploy our applications in the AWS cloud. As always, we make use of CloudFormation templates to automate the deployment, and we start by defining a VPC, virtual private cloud, to encapsulate our applications, and within the VPC we define a private subnet. This private subnet is not connected to the internet, which increases the security of our application, and the clients can log in and connect to our apps then via VPN, either through the organizations or client. We provide an S3 bucket for uploading files. Those are the point where the file servers or the FASTQ files from the sequencers are uploaded that are then triggering the ingestion of our raw data. And first we define the backend. The backend is composed of the Django app and the PostgreSQL database. For the database solution, we actually make use of a managed service by AWS, which makes the deployments, backups a bit more efficient and easier to automate. Then we have the Django application that is communicating to the PostgreSQL database. And we have a number of AWS services which we also deploy in the private subnet in order to trigger our bioinformatics steps, as we've seen before. First, we have the step function that is triggered once new data arrive in our upload bucket from our sequencer. We trigger the step function and then register and QC our FASTQ files and store the results in our database. We then have the batch queue in order to perform the RNA-Seq data processing. For the batch queues, we also need to define a compute environment, which basically sets the maximum resources that can be used and configures how the containers that make up the batch jobs should be run. And finally, we have the lambda function that is executing the differential gene expression. And these batch functions and lambda functions are triggered from the backend. We do this in order to manage permissions for certain users to trigger certain AWS services. And the user interface is deployed as a Streamlit application. The users can log into this app, and then the users are also authorized against the backend to decide if they have permissions to view certain files or projects. And we've seen in the first part how to manage datasets from the user interface, and now we will have a look at how to use the user interface to trigger RNA-Seq jobs and analyze the differential gene expression results. We are back in our user interface and we want to run the RNA-Seq data analysis pipeline. For this, we moved into the RNA-Seq page here. And first in the datasets tab, we get an overview of all the FASTQ datasets that have been analyzed and processed. And we see that this is still empty and we want to change this now. So we move into the jobs and execution tab, where we find an overview of all the RNA-Seq analysis jobs that are running on our batch queue. And to trigger a new job, we go to the Run RNA-Seq job button and find a window where we can select all the datasets that we want to analyze. So we have the option to select a genome here that we want to perform the alignment for. The species, we have different filters here. For instance, we can select only datasets that have been assigned to a certain project, like P1, which holds all our tumor and normal tissue. And now we're going to simply select all the datasets that we have in our project P1 and start the analysis. And this brings us back to the RNA-Seq analysis page, where we get an overview of all the data sets that are currently in our batch queue. This can take a little moment, and here we are back listing all the jobs that we are currently executing, when they were created, and basically their status on the job queue. We're now back in the RNA-Seq page and the datasets tab, where we have a list of all the RNA-Seq datasets that were analyzed, and we see they completed analysis now. We can check on some statistics for each of the RNA-Seq dataset files, for instance, the fraction of reads that were aligned or the version of the pipeline. We can also download the RNA-Seq quantification data or delete the datasets again. And now we want to move to the gene expression page here, where we can run the differential gene expression job. For this, we create a new job. We'll call it DGE3. And we have to define a reference and a query group. So those are just terms to name the two groups that we want to compare. And in this case, we are going to compare the healthy data sets versus the tumor. And in our case, we allocate now all these normal healthy RNA-Seq data sets that were analyzed to the healthy group and the tumor data sets to the tumor group. We can start the job here in order to trigger the lambda function, which is going to take a couple of seconds. And after we completed the differential gene expression job, we have a list of all the completed jobs here, and we can click the Explore button to plot the results in different formats. We have implemented, for instance, here a volcano plot that is comparing the fold change in abundance against the significance, against the p-values. And this helps us at the outliers to identify genes which are significantly enriched or downregulated. We can also get a tabular view of the top differentially expressed genes. This is, for instance, a list of the top upregulated genes in the tumor. And we can download the results here now also to a CSV file and use that for our target prioritization. And that was an overview of the user interface, and we're going to log out again. So we're at the end of this case study where we looked at an RNA-Seq workflow for discovery of novel drug targets, and we saw how we can build a nice data infrastructure around such a research process. If you have questions or comments, please reach out to me, and otherwise I wish you a great day.